morning, everyone. My name is Ricardo Baptista, and I'm the Chief Technology Officer of SmartCella. And the title of this talk is Innovation in Allogeneic Therapies and Targeted Delivery of Clinical Payloads. In the next 15 minutes, I hope to give you a brief overview of what we are doing at SmartCella. SmartCella is a Swedish biotech company working in developing targeted deliveries and developing regenerative medicines. And we operate in two business segments: targeted delivery with a novel endovascular delivery device that can pretty much deliver any payload into any organ by navigating through the body's vasculature. The other segment, regenerative medicines, pretty much works with two modalities. One modality of mRNA therapies, currently with a program on osteoarthritis, and cell therapies, working on allogeneic or developing allogeneic stem progenitor cells derived from human pluripotent stem cells and iPS cells, currently with two programs, one on heart failure and another one on Parkinson's disease. So just starting with the target delivery segment from our company and giving an example of what are the challenges currently in terms of delivering payloads systemically. And one example is the solid tumor treatment, where the challenges are how to deliver in the right location with the right dose and at the right time, as well as some challenges regarding hostile microenvironments in the tumors, as well as accumulation of products and subproducts in delivery. Sweden is quite renowned worldwide by its medical device development. So in the early 20s, Seldinger developed an endovascular catheter that uses the Seldinger technique, which is basically going through the vasculature and navigate through the body using the vasculature, so from outside to the inside of the body. And what we did at SmartCella, our CMO, Staffan Holmin, a neurosurgeon at Karolinska Institutet, he developed so the other way around, which is going from inside the vessels to outside the blood vessels and allow direct access to hard-to-reach tissues and organs. And that led to the creation and the development of the Extroducer, which is an endovascular catheter that allows targeted and precise delivery in pretty much any organ. I'll speak a bit more about the data that we have generated to date and how we plan to use this in our programs as well. The benefits of also of this device is that doesn't or reduces hemorrhage and thrombosis, and also the simplicity of its use make it really a low cost in terms of staff training at the hospital. So with this technology, we can reach, as I mentioned before, so hard to reach organs, tissues. We have a significant tissue area coverage, so in all the organs that we can reach pretty much any site, any area of the organ. As I said, it's easy to use. There's a reduction of the capex involved in terms of patient handling. And here in this video, you can see how simple it is to use the catheter. So what we're seeing at the moment is the surgeon is injecting or is introducing the microcatheter in a pig, and with the help of PET scan, we can actually monitor where we want the catheter to go to in terms of the vasculature. Then once the microcatheter is introduced, then we introduce the protective sheet with the needle, with the catheter per se inside. And as you can see, the thickness, it's a hair thickness of the needle, and we can, the length that we can achieve outside the blood vessel, it's between half a centimeter to 1.5 centimeters. So we have three lengths of needle. So with the help of PET scan and also a floor mark in the needle that allows to see where you are actually traveling with the catheter and the precise delivery of your payload. So we have generated data that using methylene blue to show actually the precise delivery of any payload, in this case using a chemical, but to different organs, such as in the pancreas, kidney and liver, the brain. We tested in non-human primate brains and also pig brains, and also the heart and the lung. You will see more data in the next couple of slides. So there's a whole bunch of opportunities and the strategies that we are planning with the Extroducer. So not only for cell and gene therapies and in specific for oncology treatment in solid tumors, but also delivery of pretty much any payload and use of drugs that are currently being used for chemotherapy and radiotherapy, and use the Extroducer to deliver them with higher precision in the organs instead of delivering them systemically. As well, delivery of oncolytic viruses, and also using markers for diagnostics for disease. So there's quite a broad application of the Extroducer. Currently, we are working with Xylocor on their phase 2b/3 trial of delivering AAV to the heart using the Extroducer, so we're working with them. We're also sponsoring and we're leading a trial at Karolinska Hospital for delivery of chemotherapy to the pancreas to treat pancreatic cancer. And we're also our internal program, SMART-01, that I'll speak about that in a bit, that uses the Extroducer to deliver cardiac progenitor cells to treat heart failure. So regarding the targeted delivery segment, so we are open for non-exclusive partnerships. We're looking for partners to explore different therapeutic payloads and their delivery using the Extroducer into precise location. We facilitate proof of concept studies with partners at Karolinska Institutet or a CRO of their choice, and we support filing and GLP studies if possible. So moving gears to our regenerative medicine segment, we currently have 3 products in our pipeline. Both of these, or all of these products, are derived from human pluripotent stem cells. The first one, SMART-1, which is actually a program that led to the creation of the company, and it was co-developed by us and in collaboration with AstraZeneca. It's a program, so the science has been developed at Harvard Medical Institute, or Harvard Medical School, and at Karolinska as well. And we're currently planning to go to phase 1 by the end of this year, early next year in the UK. And the product comprises of cryopreserved human cardiac progenitor cells derived from pluripotent stem cells, and we plan to use the Extroducer to deliver these cells with precision into the left ventricular through the vasculature. SMART-02 program, it's a program that was recently licensed from Johan Eriksson's lab at Karolinska Institute. The product are progenitor dopaminergic producing cells to treat Parkinson's disease, and we also plan to use the catheter for delivery of this product in the brain. The last one, SMART-03, is part of the modality of mRNA, of our mRNA platform, and uses IMSCs transformed with mRNA factors to treat osteoarthritis. I'll speak a bit about the first, SMART-01 and SMART-03 now. Regarding SMART-01, so as I mentioned, SMART-01 is cryopreserved allogeneic progenitor cardiac or human ventricular progenitor cells derived from human pluripotent stem cells. And what you can see on the left, so it's a summary or a graphic summary of the GLP tox studies. And on the bottom left, you see cells so showing that the cells engraft, they mature into cardiomyocytes, and actually they establish connection with the host tissue and promote vasculature. On the right side, it's some efficacy data, and the take-home message of these 2 plots that show 3 doses that were tested in pigs. The main take-home message is that there is no progression of the disease. In some situations, in some doses, you actually see a benefit, but the take-home message is really like the disease does not progress in terms of infarct volume and also left ventricular extraction fraction. How we plan to deliver the cells into the heart. So on your right, you see a 3D angiography model. So we use magnified or augmented reality tool to plot or to draft a model of the pig heart. In this case, in red, you see the infarcted area. We also highlight in the spheres that on the circles that you see, highlight the areas that planned for injection using the Extroducer. On the left side, you see actually what happens in the surgical theater, that the catheter, with the use of PET scan, the catheter is actually driven driven towards the injection sites, and the cells are delivered with high precision. An important feature to see that you can see on the left picture is that the catheter does not come out when the heart is beating. So there's no bleeding or no scratching of the region of the injection. So that's also one important feature of our Extroducer. Here we see that histologies of some sections of the heart of the pigs that were injected with the Extroducer, that we see that cells engraft in the pig myocardium and they mature into cardiomyocytes. So similar data that we had from the GLP tox studies. Last but not least, so our second modality in the regenerative medicine segment, we are using cells as a vehicle to deliver mRNA to circumvent challenges regarding the stability of the mRNA, the potential toxicity and challenges with using LNP particles. And currently, most of the mRNA therapies are targeted to the liver. So with this technology, we believe that we can target more organs and having a stable, let's say, active substance. And what are we using as vehicle? We are using IMSCs. So we use our IPS platform and have a protected differentiation protocol towards IMSCs. We freeze those IMSCs, then we thaw, and then we transform with different factors with mRNA, and then we have an IMSC plus the cargo to be delivered. And our first program is focused on osteoarthritis, and here what we show here is the preliminary data obtained with a rat study for 7 weeks in an osteoarthritis model. And what we can see on the left is that after 7 weeks, there's a complete alleviation of pain, which is extremely promising, the data that we generate. On the right side, you see actually regeneration of the cartilage from the osteoarthritis model. And with 7 weeks, we observed a 42% increase in cartilage volume compared to the untreated animals. So the data suggests regeneration of cartilage, and we're quite enthusiastic with this program, and we're now taking or doing a long-term study with rats. So I hope that in the past 15 minutes, I was able to give you a brief overview of what we're doing at SmartCella. And in summary, we have a targeted delivery device, the Extroducer. So please contact us if you believe that you would benefit from this device and you're looking for targeted delivery of your payloads, and also present our programs, our clinical development programs on heart failure, Parkinson's disease, and osteoarthritis. And you can reach out to us, myself, and Sabine Ott, which is the Chief Commercial Officer of SmartCella. So thank you much for your attention.